Hi, y'all. Mrs. Blasky. Last topic of section of Unit 4 today is the Beer-Lambert law. So we've been talking about how different wavelengths of light can affect molecules. This time, we're going to be talking about using different wavelengths of light, especially visible light, to help us determine the concentration of a solution. So we already know what is there, but now we want to know how much is there. Um, so this concept is based on the idea of absorption and transmission. So this is something you probably haven't talked a whole lot about in science, maybe a little bit. Um, but if somebody's wearing a blue shirt, the reason that it looks blue to us is because the molecules of the coloring of that shirt, or the molecules that make up that shirt, are absorbing all other wavelengths of light other than blue, but they're reflecting or bouncing back the blue wavelengths. So when that light gets bounced off of that shirt, it goes onto your eye, and then you can see those different colors. So if that shirt is absorbing all other wavelengths, it means it is transmitting or reflecting that blue. Normally, we talk about absorbing and transmitting when we're talking about solutions where that light can pass through it and not necessarily opaque substances like fabric. Um, so in chemistry, we have instruments that we can call either spectrophotometers or colorimeters that help us do this. Spectrophotometers are a little bit fancier. They can do more than just visible light. Colorimeters use only visible light. So inside, we're going to use colorimeters in our class. You have a light source. That light source is then sent through some sort of prism to separate out the different wavelengths of that visible light coming from that light source. And then it goes through, sometimes these are called slits, sometimes these are called diffraction gratings, that focus only to let one wavelength or a small group of wavelengths through that opening. That specific group of wavelengths is then sent through the sample. This container that the sample is in is called a cuvette. It's sort of a square shaped little mini test tube kind of thing. And then there are sensors that measure the change in that intensity of that light from before it went through the sample to after it went through the sample. So then we can determine how much of that light was absorbed by the sample, how much of it went into those molecules, and then how much was transmitted or how much came back through that sample. Now, when we do this, we can use that absorbance or how much of that light was absorbed by the sample to correlate to concentration. And so this equation is actually the Beer-Lambert law. A here is the absorbance measurement. Absorbance has no unit. It's usually a decimal point number that we could turn into a percentage. This weird little E symbol is an eta, is the molar absorptivity, which is a constant value for a given substance. It helps describe how intensely the sample absorbs light of a specific wavelength. So this is not only sample specific, but also wavelength specific, but it is a constant for those. B is the path length. That's how long that light traveled through the sample. So that has to do with the size of your cuvette. And then lowercase c here stands for concentration in molarity, so that we can see that our absorbance and our concentration are going to be directly proportional. Most times when we do Beer's law experiments, the cuvette size is standardized to 1 centimeter. So that we always just sort of assume that that B is 1 unless we are told otherwise. When we do these experiments, we need to, not only do we need to put the sample that we want to measure inside of that machine, but we also need to calibrate the machine, which means we need to teach that machine what 100% transmission and what 100% absorbance are, so that the instrumentation itself, the colorimeter, can figure out, is any of this absorbance that I'm measuring coming from just the cuvette, the plastic of the cuvette? Is it coming from the solvent for my solution. And so we have to make this thing called a blank. A blank is a cuvette full of just the solvent for our solution. So if we're dissolving things in water, we'd put a cuvette just full with pure water in there. So that we can teach the machine, this is what a zero concentration solution looks like. So any absorbance that you're getting from the solution, we want to sort of subtract that out because we know it's not coming from the solute that we're trying to measure. Because these instruments are rather specific, because they can measure these things fairly accurately, any sort of small error can affect our results drastically. And this is something that AP questions like to ask you about what could have caused this error. Two things that we usually talk about as causing errors when we read absorbance is a change in concentration. Because our sample is so small inside of this cuvette, a drop or two of extra water inside that cuvette from cleaning it out can change the concentration enough to change the absorbance. So instead of rinsing it out with water, when we're done, we're going to rinse with water a couple times to make sure it's clean. And then we're going to do a rinse with the solution itself to make sure that there's no drops of water left inside that cuvette to change the concentration. Another reading error that can come from this is touching the cuvette too much with your fingers. If you leave fingerprints on the outside of the cuvette, the oil deposited from your skin can actually affect the results also. So water inside the cuvette can decrease the concentration, which would decrease the absorbance. Oil deposited on the outside from your fingerprints would block the light from going through, so that would increase the absorbance. So just to show you real quick, this is what our colorimeters are going to look like in lab. Doesn't look super fancy, but here is that square plastic cuvette down in the colorimeter. Inside of this machine, we've got the light source and the prism shooting the light through here. That then sends it to the detector, and that detector allows us to be able to read those values on our computer screen. The other thing that we need to think about when we are doing these samples and reading Beer's Law calculations, or reading to be able to do Beer's Law calculations, reading these absorbance calculations, is the color of the solution and what that tells us about what colors of wavelengths of light the sample absorbs and which ones it will transmit. So in this case, this sample looks sort of reddish orange to me, which means that we would actually want to measure this sample in green wavelengths. Because if this sample is reddish orange, that means that it is absorbing all of the other wavelengths, but only transmitting or only reflecting those red-orange wavelengths back to us. So interestingly, samples absorb best at the total opposite color, if you think about your color wheel, from the color that we see. So if we see a red sample, we know that it's going to absorb best in the green range. If we see a blue sample, we know it's going to absorb best in the orange range. If we see, what color am I missing? A purple, then we know that it's going to absorb best in the yellow wavelength ranges. For our colorimeters, they're not super accurate in what wavelengths they can produce. They definitely produce in ranges of wavelengths due to colors. So in class, we are going to go over some practice problems about how to analyze these. The thing that we see the most often for Beer's Law calculations on a problem is what we call a standardized curve or a Beer's Law graph, where we would take different known molarities of a sample, measure their absorbances, and we can create that straight line graph. If we can find the slope of that graph, that slope is that eta, that molar absorptivity number in our Beer's Law equation. Then we can either use the equation or we can use the graph to help us identify the concentration of an unknown. So let's say we had this particular graph and we put an unknown solution into our colorimeter, and we found out that we had an absorbance right around 4.5. we say, well, an absorbance of 4.5, follow that graph over here. That means our concentration of that sample would be really close to 0.08 molar cobalt ion in this case. So by setting up the standard curve, by setting up the standard line, we know how concentration and absorbance are related to each other. And so then we can make that relationship known for any unknown solution of the same chemical. Now, once we moved away from cobalt, maybe instead of cobalt, we had copper or iron or something else, we would have to create a totally new standard curve for that different substance. So we will be able to do a lab with this in class so that you can get a better handle of how to do all this and how to do all the math. And that should help you understand a little bit more about Beer's law. I'm done for now, and I will talk to you later.